Okay, the various factors that change the or that affect the enzyme catalyzed reaction. So what factors come in your mind? Yes, Amna, which you did in O levels or IGCSE that affect the rate of enzyme activity?
It's the amount of substrate present,
Okay.
the amount of product formed, and the rate at which the product and substrate are being formed and used up.
And besides that, limiting factor, any limiting factor that can increase or that can decrease the speed of a reaction? What are those? Does temperature affect the rate of a reaction?
Yes, miss.
Does pH affect the rate of a reaction?
Yes, initially it does not, but as the reaction progresses, it does.
Okay. So now this question is for both of you. You can see, I'm actually zooming into it a lot. You can see a graph that is showing you the progress of an enzyme catalyzed reaction over a certain temperature range. It seems as if the reaction is slowly and gradually beginning. It increases as temperature rises and then it falls down. On the x-axis, you can see temperature plotted in degrees Celsius. On the y-axis, you see the rate of reaction that has been plotted. No specific units have been given to it. Let's consider these units to be arbitrary units. Arbitrary ka matlab imaginary units. Okay. Can I say roughly, and this question is both of you, that as I'm increasing the temperature by 10 degrees Celsius, the rate of the reaction is being doubled? Yes or no?
Yes, miss.
Did you understand my question? My question is that as the temperature of the reaction is being increased by 10 degrees, हर 10 degree rise होने के बाद, is the rate being doubled as compared to before?
Yes, miss.
It is. See, 0 to 10. At 0, the rate was at this place, and at 10, the rate is at this place. So almost double. Then let's see the difference between 10 and 20. 10 पे यहाँ पे है, 20 पे यहाँ पे है. So is this double? Is the rate double?
Yes, miss.
Right? And so on. So we keep seeing that as the temperature is increasing, the rate of the reaction is doubling till finally, when the temperature, which is 40 degrees Celsius, we can see that the rate of the reaction is maximum. So since it's reaching a peak, so we can say that 40 degrees centigrade or Celsius is the optimum temperature for this reaction. And when I say optimum, I mean that it is that temperature where the maximum number of collisions are taking place between the enzyme and the substrate. All the active sites of the enzyme are completely occupied. Jitna enzyme ko kaam karna tha, wo kar raha hai. In other words, we can say that the enzyme is working fastest at this temperature, सबसे ज़्यादा, क्योंकि सबसे ज़्यादा maximum rate यहीं पे ही है. So the enzyme is working fastest at 40 degrees Celsius because the rate is the maximum. Beyond 40 degrees Celsius, if we keep on increasing the temperature from 40 to 45 to 50, 55, we can see that the rate is dropping. And the rate is dropping because it shows over here that the enzyme has become denatured. It keeps dropping till finally it reaches around 55 degrees Celsius पे जो है वो आपका rate कितना हो जाता है? 0. ठीक है? So why is it 0? Because this is that place when we say the enzyme has completely become denatured. Acha. What do you mean by denatured? Yes, Amna.
Yes, the enzyme loses its shape, therefore the active site is deformed and the lock and key mechanism is no longer in work.
Enzyme loses its shape. Okay. Structurally, which biological molecule is the enzyme? Yes, Amna.
Yes, I didn't understand the question.
Structurally, an enzyme is what biological molecule?
Protein.
Protein. Okay. So if I say the protein has become denatured, can you explain this phenomenon to me at the level of the bonds, keeping in mind that this protein has a tertiary structure? If I heat a tertiary protein or any protein that has a tertiary or a globular structure, what is going to happen at a chemical level, at an atomic or a molecular level?
This I think the bonds that make up the enzyme, the bonds they are going to be broken down.
Okay, I think this answer can be better told by Alia. Alia, you have done biological molecules with me in great detail. So tell me one thing,
Uh,
Alia. Is enzyme a globular protein?
Yes, miss, it's a globular protein.
Acha, if it's a globular protein, does it have a tertiary structure?
Yes, miss, it does.
Tertiary. When I say it has a tertiary structure, what do I mean when I say it has a tertiary structure?
Miss, it has more than one polypeptide chain.
No, not at all. That is not the meaning of tertiary. That's the meaning of quaternary.
Miss, it has the hydrogen bonds and then the
And
disulfide bonds.
and
Miss, the hydrophobic bonds as well.
exactly. When I use the word tertiary, what I mean is that it's a protein. No, sorry, it's a kind of a globular protein which has all four kinds of bonds. Can I say now? See, I have to connect this with the previous knowledge gained. Look, by by coincidence, this slide has opened. An enzyme is a globular protein. Remember, Alia, हमने ये slide की थी. A globular protein folded into a ball shape. The reason it's folded into a ball shape is because of presence of those four bonds. ठीक है? अच्छा, and when a protein becomes ball shaped like this, तो क्या होता है आलिया? The hydrophobic groups, R groups are pointing where?
Miss, they point inside while the
And-
hydrophilic groups point outside.
Okay, why do the hydrophilic groups point outside?
Because miss, they are soluble, so they can dissolve substance.
Okay, so do you think enzymes are water soluble?
Miss, they react with substances, so
They
then
are,
they can.
of course, they are water soluble. They are water soluble, right? Do you think the shape of the enzyme is their key to their function?
Yes, miss.
Now look here. दोबारा से हम इसको connect करते हैं. We are connecting this chapter with enzymes. We did globular proteins in biological molecules. We said a globular protein molecule is the one which is folded into a globule, जो sphere shape होता है. Why is it folded? Because of presence of those four kinds of bonds. ठीक है? सारी globular proteins जो होते हैं, they have hydrophobic groups in the center and they have hydrophilic R groups on the outside. Hydrophilic R groups are facing the aqueous environment of the body. Example जब हम देते हैं किसी globular protein का तो हम क्या कहते हैं? Example is enzymes, antibodies and hormones. ठीक है? The shape, the globular proteins have a very specific shape and their shape is the key to their function. Okay, Alia, this question is for you. Do globular proteins have a functional role or do they have a structural role?
Miss, they have a structural role. Yes.
Think about it. What? Read this slide once again. A globular protein, you are saying it has a structural role. What do enzymes
Miss, for
do?
example, because miss, for enzymes, they have the specific active site which is complementary to the substrate. So
नहीं,
would that be structure?
no. Structural role का मतलब आपकी body का कोई structure बनाता है वो. Enzyme doesn't make a structure of your body. Like collagen makes the structure of your body, which is the wall of the blood vessels or the tendons of the muscles. ठीक है? Cellulose जो है वो भी एक structural biological molecule है क्योंकि वो cell wall बनाता है, right? Enzyme is not a structural molecule. It is a functional molecule. Why? Because enzymes catalyze chemical reactions. समझ आ रही है बात?
Okay.
Right? I think you guys need to, revise from whatever I have been doing with you, especially in biological molecules. इसका तो मुझे test लेना पड़ेगा. इसका next, can you be prepared for a test on biological molecules next week, both of you?
Yes.
ठीक है?
Yes, miss.
Please be prepared. वरना ये अगर आपको if you forget the very basic concepts of biological molecules, तो आपको problem होगा बहुत ज़्यादा. सबसे ज़्यादा difficult question इसी chapter के आते हैं, right? For example, Alia, when I was doing this question with you, I deliberately gave you these two questions. What do you mean by a globular protein? Explain the biological significance of globular protein. So the main biological significance of a globular protein is that it's functional. ठीक है, वो function के काम आता है, structural नहीं है वो. Look, globular proteins always have a function. They can, ज़्यादातर globular proteins या तो enzymes होते हैं या hormones होते हैं. Hemoglobin itself is a globular protein because it has a function to transport oxygen, or antibodies are also globular proteins, right? Coming back to where we were on enzymes. और हम क्या बात कर रहे थे? Sorry. हाँ. We were talking about, that's why I had to, go back to the concept about enzymes, because in paper 3 also and in paper 2 also, the examiner actually wants you to write down about क्या मतलब है इस बात का कि enzyme becomes denatured. So an AS student should very clearly know what is the meaning of the statement enzyme becomes denatured. इसका मतलब ये है कि you will talk about all the bonds. You will say enzymes are, अब मैं कुछ, I'm just narrating some points and both of you will remember these points and then again repeat it, okay? So I will say the enzyme becomes denatured because it is a globular protein. It has a tertiary structure. Extremes of temperature will denature it because it breaks the hydrogen bonds. When the hydrogen bonds are broken, the enzyme loses its three-dimensional configuration and hence loses the active site. When it loses active site, the rate of reaction falls, right? Yes. Amna, repeat what I said right now.
Since it's a enzyme enzyme has a tertiary structure, once we give it excess heat, more than what it needs, the tertiary structure is broken down, the bonds are broken down, and therefore the active site gets deformed and it can't function anymore.
It can't function and the rate drops, right? Acha. Now this kind of graph can be given to you, and this is the most frequently asked question in exams. You can be asked to describe the graph or you can be asked to read the graph. Okay? What is the difference between describing the graph and reading the graph? Yes, Alia.
Miss, for reading the graph, we basically relate it to the scientific concept, miss, and then we make use of the values. But when we are describing the graph, we focus more on the shape of the graph.
Sorry, I know I used the wrong word. What is the difference between describing and explaining the graph? Describing is actually just quoting data. अगर मुझे describe करना है, let's say अगर यहाँ पे भी numbers होते ना, if there would have been numbers over here like 1, 2, 3, 4, 5 and 6. तो अगर मुझसे कोई पूछता कि describe the graph, तो I would have said कि at 10 degrees Celsius, the rate of reaction is 1 unit. At 20 degrees Celsius, it's 2 unit and so on. तो ये मैंने describe कर दिया, right? If I'm asked to explain the graph, then I will explain it like this. I have a graph over here. This is how I explain the graph. ठीक है? For example, this part, number 1. If I am asked to explain this part, तो मैं क्या कहूँगी? I will say, at temperatures of 0 degrees Celsius, the enzyme is inactive, right? Since it's inactive, so there is no chance of the enzyme interacting with the substrate, and hence no enzyme substrate complex formation takes place, and therefore the rate of the reaction is minimal. हो सकता है कि मुझे एग्जामिनर कहे कि एक्सप्लेन दिस पार्ट ऑफ द ग्राफ, यानी यहां से यहां तक, जिसको मैंने नंबर 2 करके लेबल किया हुआ है. सो आई वुड से that as the temperature is increasing by every 10 degrees Celsius, the rate of the enzyme catalyzed reaction is increasing because increased heat energy is giving increased kinetic energy. Increased kinetic energy is increasing the frequency of the collisions between the enzyme and the substrate, and hence more enzyme substrate complexes are formed and hence the rate of the reaction is increasing. ठीक है? एग्जामिनर मुझसे ये भी पूछ सकता है कि एक्सप्लेन दिस पार्ट ऑफ द ग्राफ, एग्जैक्टली ये जो पॉइंट नंबर 3 है, दिस पार्ट. सो आई वुड से दैट द एंजाइम एक्टिविटी इज मैक्सिमम at point 3 because that's the optimum temperature. Optimum temperature meaning this is that temperature where maximum number of the enzyme substrate complexes have formed and the enzyme is working at its fastest velocity or rate of reaction. फिर मुझसे कोई पूछ सकता है कि आप ये वाला पार्ट एक्सप्लेन करें, पार्ट 4. सो आई विल से दैट once the temperature goes beyond 40 यानी 40 से ज्यादा बढ़ने लगता है टेम्परेचर, see what's written over here. An AS student, अगर कोई AS का स्टूडेंट है ना, अगर वो सिर्फ ये लिखता है कि the enzyme has become denatured explanation में, उसका मतलब कि उसने पूरा आंसर नहीं लिखा. You are expected to write like this. That's why I went back to the slide of biological molecules. ठीक है? You're expected to write down कि secondary structure टूट जाएगा. Secondary structure में hydrogen bonds होते हैं, तो वो जब खत्म हो जाएंगे तो tertiary भी खत्म हो जाएगा, right? The enzyme will unfold. पहले तो enzyme ऐसे fold हुआ था. Now the enzyme will be like this, unfold because of loss of hydrogen bonds. When unfolding takes place, the precise 3D structure is lost and hence the active site is also lost. उसी की ही वजह से rate जो है वो आपका कम हो रहा है. अच्छा, you can mention about breakage of hydrogen bonds also. और आप हाइड्रोफोबिक बॉन्ड्स के बारे में भी कह सकते हो. एग्जामिनर दोनों को करेक्ट समझेगा बिकॉज जब आप हीट करते हैं एंजाइम को तो द हाइड्रोजन बॉन्ड्स आर ब्रोकन एंड द हाइड्रोफोबिक इंटरैक्शंस आर आल्सो ब्रोकन टिल फाइनली व्हेन the temperature is 55 degrees Celsius, the enzyme is completely and irreversibly damaged, denatured, and hence the enzyme activity is 0. G, is this understood, both of you? दोनों को अच्छी तरह समझ आ गया? Any questions from this? No questions?
It's clear.
Clear? Let's move on to the pH wala part. Acha, okay. Go through these bullet points quickly and then I'll move to the pH part. When you're done, let me know. The effect of pH on the rate of a reaction. So, Amna, you tell me, by looking at the graph, what do you think is the optimum pH for this particular reaction?
7.
7, right? Most probably it looks like and the enzyme salivary amylase because salivary amylase is in the saliva and saliva has a pH of 7. It's a neutral pH. If this graph was for the stomach enzyme, which is pepsin, the peak reaction would have been between 1 and 3 because pepsin works at acidic pH. Trypsin is an intestinal or a duodenal enzyme और उसका जो pH है वो 7 से ज़्यादा होता है. Optimum, it's sort of alkaline. अच्छा, if you want to explain the effect of pH, कि pH पे क्यों ऐसा होता है कि कुछ enzymes किसी pH पे काम कर सकते हैं और कुछ किसी पे. So in that case, you will be talking about not hydrogen bonds, but you will be talking about ionic bonds. If you remember, when I was teaching you these 4 different kinds of bonds, जब हम proteins पढ़ रहे थे, so I told you that some of the R groups can be charged, okay, charged R groups. So if you have 2 charged R groups facing each other, ionic bonds can automatically form between them. When you alter the pH, you are altering the concentration of hydrogen ions. जब pH low होता है, तो hydrogen ions की concentration बढ़ जाती है. जब pH high होता है, तो hydrogen ion concentration becomes less. So depending upon what the concentration of hydrogen ions is, it affects the ionization of the R groups. और जब आपकी ionization R groups की affect होती है, तो वो भी affect करता है enzyme के shape को, right? जैसे for example कि अगर trypsin की हम मिसाल ले लें, trypsin is the enzyme that works best at a pH of around 7.9, ठीक है? हम अगर trypsin को एक acidic environment दे दें, तो the active site of the trypsin will be lost. Why? Because when we lower the pH, we are increasing the acidity. and hence we are increasing the concentration of the H plus ions. उससे होगा ये कि ट्रिप्सिन जो मॉलिक्यूल है प्रोटीन का उसके अपने R ग्रुप्स की आयनाइजेशन चेंज हो जाएगी because of which it will lose its 3-dimensional shape or configuration. वो अगर folded shape का था तो वो ऐसे हो जाएगा, straight हो जाएगा, it will lose its active site, right? So here दो तरह की चीजें हैं, ठीक है? You can be asked, आपसे ये पूछा जाएगा कि why does an enzyme lose its active site when it is exposed to extremes of temperature or when it is exposed to extremes of pH? So when the question is on extremes of temperature, you are going to talk about hydrogen bonds and hydrophobic interactions. And when the question is on extremes of pH, you're going to talk about ionic bonds. ठीक है? Ionic bonds की जो discussion है, it's on this slide, and both of you can read this. And if you don't understand anything from this slide, you can ask me. What is plotted on the x-axis? Anyone can answer. What's plotted on the x-axis? Keep yourself unmuted, both of you. Yes. What is plotted on the x-axis?
This is the concentration of the substrate.
Substrate. What is plotted on the y-axis?
This is the initial velocity.
Initial velocity. Acha. Is the substrate concentration changing in this graph as time passes?
Yes, miss, it's changing.
It's changing. Very good. Acha. Look at this slide of mine very carefully and tell me, as far as the other conditions are concerned, is the enzyme concentration constant?
Yes, miss, it's constant.
It's constant. अच्छा. ये आपको कौन सा reaction उन्होंने दिखाया हुआ है? ये same reaction है in which hydrogen peroxide H2O2 is actually broken down by the enzyme catalase into water and oxygen. If you guys have in case forgotten, I'll repeat once again that hydrogen peroxide is produced inside the human body as a result of some reactions. और बहुत ही dangerous toxic substance है ये. But we have provisions inside our liver, which is the enzyme catalase, which can very quickly, within seconds, it can break millions, literally millions of molecules of hydrogen peroxide into water and oxygen. Okay? And this is how the body gets rid of such a toxic substance. अच्छा. अब जब ये reaction होता है, तो our substrate is H2O2. Okay? ये जो आपका substrate लिखा हुआ है ना, ये है H2O2. It is broken down into water and oxygen और enzyme का नाम है catalase. अब जो ये substrate है ना, ये हमारा बहुत काफ़ी है हमारे पास because as we are moving from left to right, the substrate concentration is increasing. Enzyme हमारे पास constant concentration का है और जो product बन रही है वो क्या बन रही है? Oxygen gas बन रही है, right? अच्छा, Amna, this question is for you. Why has the initial velocity of the reaction being plotted on the y-axis and why not the entire velocity? Initial velocity कोई क्यों plot किया हुआ है यहाँ पे? Any idea?
Just because the initial velocity is not affected by the temperature or pH, so we can get more accurate results.
Very good. And this should be your answer, okay? Whenever the examiner asks you, why is the initial velocity plotted? You will say, during the initial 30 seconds, the different factors, they have not affected the enzymes and the substrate. So that will give you a more accurate representation of the reaction. ठीक है? अच्छा, अब हो क्या रहा है यहाँ पे? I am now going to explain this graph to you, okay? As I increase the substrate concentration, the initial velocity keeps on increasing till finally, maybe I can say that during this time period, there is a sort of a kind of exponential increase. And then finally, the rate slows down till a time comes when the reaction reaches a plateau. Any idea कि ये plateau क्यों reach हो रहा है? Any idea? Alia, Amna? The answer is found on this slide only. Why does the graph reach a plateau?
Miss Lato, can you repeat that word?
Plateau. Acha, sorry, I should have told you the spelling. The plateau. Or in other words, I can say, let me use simple words. Why does the graph flatten out? Plateau का मतलब होता है flattening out. It is flattening out, right? Straight हो रहा है. तो ये क्यों हो रहा है? Any idea? At this point.
Just because now the limiting factors are starting to take effect, so the rate
Which
is
limiting
starting
factor? Which limiting factor? What is that factor that is limiting the rate of the reaction at this point?
This the amount of substrate concentration.
How can you say substrate? Substrate तो हम constantly बढ़ाते जा रहे हैं, substrate तो हमारे पास बहुत है. What's the limiting factor?
This the number of active sites then.
Exactly, यही तो लिखा हुआ है ऊपर, right? In the beginning, the condition that we kept was that we had a constant concentration of enzyme. ये limited concentration है, ठीक है? हम वो जितनी भी हमारे पास थी वो constant ही है. तो जाहिर है एक वक्त आएगा when all the active sites of the enzymes will be saturated. So that's that point where the graph flattens out. इसको हम use करते हैं flatten out या it plateaus. You also have to use the same word. It plateaus. So why does it plateau? Because all active sites of enzymes are saturated. We have plenty of substrate, but it's of no use to us. ठीक है? Whenever you see such kind of a graph in which the velocity of the reaction does not increase beyond a certain point, we say, ये वाली point है ना, ये वाली point, we say that the enzyme has reached a maximum velocity. A maximum velocity. और इसको हम medical language में या biological terminology में हम कहते हैं Vmax. Vmax stands for maximum velocity. So again, I'll repeat once again. The graph which you guys are looking at is a typical graph that shows the effect of increasing substrate concentration on the initial rate of reaction. Initially, the rate increases, but then the rate slows down till finally it reaches a plateau beyond which, no matter how much you increase the substrate concentration, the rate will not increase. वो ऐसे ही रहेगा जिंदगी भर. Why does it happen? Because we have a limited amount of enzymes. All active sites of the enzymes are saturated. This point, exactly this point beyond which the velocity cannot increase is known as the Vmax or the maximum velocity. तो हम ये कहते हैं कि this is the maximum velocity with which the enzyme can work. Beyond this, इससे ज़्यादा velocity नहीं हो सकती है. ठीक है? We can't have a maximum velocity more than this. Then there is another point which has been plotted over here, which is known as half Vmax. Half Vmax का मतलब कि ये dotted line वैसे यहाँ पे गलत बनी हुई है, इसको यहाँ ऐसे बना हुआ होना चाहिए था. It should have been like this. Okay, wait. This is a Google image, so Google images कभी गलत भी होते हैं. So this line should have been plotted like this. ठीक है? Exactly the half of this line, यानी कि यहाँ पे आपका Vmax आता है तो half Vmax यहाँ आता है. So this is half Vmax. अच्छा. The concentration of the substrate at which the enzyme is working at half its maximum velocity is known as Km. Km. Okay? Km is the concentration of the substrate at which the enzyme is working at half its maximum velocity. Now, what will I do? I am going to go back to the textbook and I want you guys, both of you, to go through the description of this. Ye wala. First read this and then I'll keep scrolling and then we can discuss all the queries. When you're done reading, let me know.